Hello friends, welcome back to my channel Medicine of 101. Today we'll talk about type 1 hypersensitivity part 2. Okay, before starting the video, let me tell you I have already uploaded the video where I have described about the type 1 hypersensitivity part 1. Okay, there actually have discussed how the Ig antibody that will be formed. Okay, so from the first part of the video, I'll just repeat again once again. The very briefly I'll just repeat. So whatever thing we saw over here to produce the Ig antibodies. Okay, now for example, the lungs over here, very rapid recap. Okay, now whenever the pollen grains are coming over here, see for example the pollen grains are coming within the lungs to the bronchial trees. For example, is the macrophage over here. Then what? This macrophage that will take basically the class 2 MHC molecule that will take. For example, let me show with this color over here. This is the macrophage. Within the macrophage, we know what? There is a presence of the class 2 molecule MHC, class 2 molecules, okay, and there is also presence of what? The receptors for CD4. Okay, let me write it over here. Then it will represent these things to the what? To the T helper cells. Okay. After that, we will see there will be interaction in between them. There is what? CD4 positive receptors over here. Along with that, there is a receptor for what? MHC, class 2 MHC, class 2 molecules. After that, we will see this the T helpers cell that will secrete the different kinds of interleukin 4, interleukin 5, and that will activate the B cells. And whenever the B cells that will be converted into the plasma cells. Now it will secrete that what? The antibodies. Here we got Ig antibodies, and I already said you that this Ig antibodies that is having the receptors what? Within the mast cell. Okay, so for example, this is the mast cell, and it is having the receptors for Ig antibody. Now the Ig antibody will come over here and it will fix over here. Ig antibody will come and it will fix over here. Okay, that's the thing I already talked about in part 1 of the video in terms of the type 1 hypersensitivity. Okay, now we'll talk about the mast cell. Okay, now at first we need to know where can we see the mast cell. So first we need to know in terms of the location of the mast cell. See, so basically we will see the mast cell. More concentration of the mast cell will get where? Around the perivascular connective tissue. Where? Around perivascular connective tissue. Vascular connective tissue. For that thing, let me just draw a little bit of the circulations. For example, is the general circulations. Perivascular means around the circulation. That means we will get over here. This is the mast cells. This is the mast cell over here. And the same type of the cells we can get where? Within the circulations, they are the basophils. So these are the basophils. Same type of the cells. The basophils, they are also having the receptors for Ig antibodies. So whenever the basophils are within the circulation, they are known as basophils. Whenever the basophils they are coming outside the circulation, that means that whenever they are in the tissue, we have to call them in terms of the mast cell. Okay, so this is the perivascular connective tissue. So perivascular connective tissue will get over here. Along with that, we will get over in the subepithelial, or we can say in terms of the submucosal lining. Here we can get in terms of in terms of the GI, you can get it over here in these locations. In the respiratory systems, you can get in the urogenital systems. We can get also in these things in the linings. In where? In submucosal lining. Okay, we'll get what? The mast cells. Oil concentrated mast cell. We'll get over here. Now, if I just show you, okay, let me use this part. I have to get some part from here. And what are the factors that can activate the mast cell? Now we have to know what are the factors or what are the components that can activate the mast cell. Okay. So, for example, is the mast cell. What are the factors that can activate the mast cell? So, first I have already talked about in terms of the what? IgE antibodies, okay, that can activate the mast cell. First thing, remember in this way. Now we can remember in this way that there are certain types of the drugs that can also activate the mast cell. What are the drugs? For example, the codeine, morphine. List of the drugs, they can also activate the what? The mast cell. Different types of the drugs over here. Then you have to know in terms of the snake bite or the venom. Snake bite or the venom, they can also activate the mast cell. Okay. Along with that, we can get over also that first I did talk about the IG antibodies that can activate the mast cell. Then I did talk about the different kinds of the drugs. For example, the codeine, the morphine, they can activate the mast cell. The snake bite or the venom, they can also activate the mast cell. And different kinds of the complementary proteins during the during inflammatory process, we get the different kinds of the complementary proteins C3A, C5A, C4A, especially C3 and C5A. They can also activate the mast cell. Apart from these things, there are also certain types of the factors that can also activate the mast cell. For example, the cold water or the cold environment, or you can say the hot environment, or you can say the physical trauma. This can activate also the mast cell. Okay. Now, physical trauma, the coldness or the hotness, that can also activate the mast cell. You can also say that strong sunlight. This can also activate the mast cell. Okay. Now these are the factors, the different kinds of the factors that can activate the mast cell. Now whatever the thing or changes we will see whenever the mast cell that will be activated within the lungs. That thing, let's draw a lungs. Okay. Okay, see, this is the bronchial trees. You get the different kinds of the branches of the bronchioles over here. In the same way, you will get over here also. Now, where we will see exactly the mast cell, another mucosal lining, so we will get the mast cell around this person here in this species, we will get the mast cell. Now, we need to know a little bit of the histological aspect from the diagram. So if I just draw from this diagram, whatever the thing will get over here, we know what with the innermost layer will get what whatever the thing will get, definitely the epithelial cell layer will get. So first, for example, we got the epithelial cellular layer, or that thing let's draw. We'll get what? The epithelial cellular layers. Epithelial cellular layers will get over here, and in the mid we'll get what? The glands. We'll see the formation of the gland. Okay, we'll get the gland. Now, after that, whatever the thing we'll get over here. At first we got these things, then we'll get around this figure, we'll get the blood vessels. Around this figure we'll get what? The blood vessels. We'll get the blood vessels. Okay. Now, after the blood vessels, we'll get what is smooth muscles, the bronchial smooth muscles over here. So if I just show these smooth muscles with this color, this way, okay. Let me show this way. Okay, we'll get the smooth muscles. Okay. First we got the what? Epithelial cell layers, then we got the glands, then we got the what? Different kinds of the blood vessels over here, smooth blood vessels, then we got the smooth muscles. Now, basically we got the what? These are the pollen grains. And these are the pollen grain antigens. These are the pollen grain antigens. Okay. Now, try to understand from here that whenever we are just talking about in terms of that, whenever we get the, IG antibodies, after the pollen grains, we'll see what when the pollen grain will come after the different types of the process. What I have already explained in the part 1 video, we'll get the productions of the what IG antibody. And now see here is the presence of the mast cell. There is the presence of the mast cell. Perivascular connective tissue in these locations. Okay. I have just shown these portions, for example, these portions largely over here. Okay. Now I will show one mast cell over here, and whatever the clinical manifestations we can get over here, we'll talk about those things. Okay. Now let's talk about the mast cell. See, if I just say, for example, this is the mast cell. Okay. And within the mast cell, there is the receptors for different kinds of the what? IgE antibodies. We'll get the receptors for IgE antibodies. Okay. Now around it, these are the receptors for IgE antibody. We'll get the IgE antibody, the receptor for Fc portions of the IgE antibody. Okay. We'll get what over here. Okay. Now see. Now whatever thing we'll get, at first the pollen grains they will come over here, and the pollen grains they will be present over here. Okay. Now whenever the pollen grains they will come over here, after that we will see there will be the cross linking of in between the IgE antibodies. So whenever we are getting the cross linking of the IgE antibodies, definitely after that we will get whatever the thing, there will be on signal that will be produced over here, that is intracytoplasmic signal. So for example, is the intracytoplasmic signal. Intracytoplasmic signal. Now whenever the signal will come, we will see within the mast cell, we will see the presence of the vesicle. Within the vesicle, we will see there is preformed chemical substances. We will see the cytoplasmic vesicles, we will see there is preformed what? Preformed chemical substances. After that we will see whenever this signal will come over here, we will see these vesicles that will get fused with the membrane, and it will secrete this type of the what? Chemical mediators. Whatever the preformed, these are the preformed chemical mediators, they will secrete out. So preformed, whatever the preformed chemical mediators will get over here, basically we will get over here that histamine. We'll get histamine. Then we will get over here the protease, and we will get neutrophil chemotactic factor, and we will get eosinophilic chemotactic factors here. Okay. Now first let's talk about the histamine. Whatever the thing will get under the histamine. So histamine, what they will do over here, what is the function of the histamine? If we say the first thing, histamine they will do what? They will go around the blood vessels, they will go over here and they will cause vasodilations. First they will cause the vasodilations. Okay, let me show over here. They will cause the vasodilation. First we will get the vasodilation. The second thing, what type of thing will get? They will just do the shrinkage in between the endothelial cells. For example, is the endothelial cellular layers over here. Let me show these things over here. Okay. This is the normal endothelial linings. Okay. Now, whenever the histamine will come over here, they will just do the shrinkage. That means the endothelial cellular shrinkage over here. As a result, we will see the permeability of the blood that will be increased. So we'll see over here in a nutshell we can say that will just increase permeability. And whenever the blood is coming from here in these spaces, in the in these portions, there will be formation. There is a chances for formation of what? Edema. Okay, there will be chances for formation of edema. So the permeability increase. Along with that, we are getting the vasodilations. So you can think like this. Okay. So the permeability that will be increased. Then you can say the vasodilations. Over here, along with that, we will see in the smooth muscles, we will see the vasospasm. That means they will constrict. They will see the constrictions of the smooth muscles. So we will see vasospasm. So we will get the constrictions of the smooth muscle. Now try to think from here that whenever we are talking in terms of these things, first we are getting the formations of the edema. So we are getting the edema. Whenever the edema is formed, definitely they will just press these tissues. That means as a result we will see the lumen that will be narrower. The lumen that will be narrowing. Why? When you try to understand, whenever the edema is formed in these regions, okay, whenever the edema is formed, they will try to compress this near or neighboring tissues. The edema, they will compress the neighboring tissues over here. As a result, we will see there is a chances for reducing the size of the lumen. Along with that, we are getting what fasciitis present. That means these smooth muscles, the bronchial smooth muscles, they are trying to contract. They are trying to contract. So whenever we are seeing the constrictions of the, whenever we are seeing the constrictions of the what? Bronchial smooth muscles, as a result, we will see further there will be what? Decreasing of the lumen over here. So ultimately we will get over here the size of the lumen. That means this space that will be very less. Now try to think, whenever the size of the lumen is less, definitely there is no sufficient space for the air passing, so there will be a problem in the breathing, especially during expiration. Okay, during expirations, the individual will face too much problems over here. Okay, now we'll talk about that what are the other complications we can get over here. Okay, now we'll talk about this over here. Around that protease functions. What are the functions of the protease you will get over here? First things, they will go cause the further tissue damage. Already damage is over here. They will cause the further tissue damage. So you can think about this thing. What are the conditions we will see over here? The second thing, they can just break down the complement proteins to produce the C3A and C5A. And whenever these things are produced, try to understand, whenever these things are produced, the C3 and C5A, for example, these are the receptors for C3 and C5A. Whenever these are produced, they will go over here, they will act over here, and they will cause the further stimulations of the mast cell. So further stimulation of mast cell. So try to think from here, that will get the amplifications. Okay, whatever the problem we'll see over here. And in these cases we'll get what, that there will be what, accumulations of the more amount of the neutrophils, more amount of the eosinophil in these regions. So we'll get more amount of the neutrophils and we'll get more amount of the eosinophil in these regions. Okay, and we'll talk about these things, whatever the effects of the eosinophil we'll get over here. Okay, in the subsequent part. Now for the first signal, this is the intracytoplasmic signal. Whenever it is coming here, it is what? It is just making the vesicles to secrete it out. Whatever the components, we can see in terms of the preformed chemical mediators, they are coming out. So this type of problem we are getting for the first times. Okay. Now this is the functions for this time we are getting over here. Now we'll talk about the second type of the signals. Now this signal that will go through the intranuclear signals. So this signal that will come over here within the nucleus. And this is the second time, whenever the nucleus is getting activated by these signals, it will produce certain types of the chemical mediators or the proteins. It will first produce the mRNA, you know these things, right? The mRNA will produce, after that it will do the translations and it will produce certain types of the proteins. These are the soluble proteins. So here, soluble proteins. And these proteins will help to interact in between the cells. These soluble proteins, let me show over here. These soluble proteins, they will help to interact in between the cells. Whatever the soluble proteins will get over here, we'll get the TNF, tumor necrosis factor, we'll get interleukin 2, we'll get interleukin 3, interleukin 4, interleukin 5, interleukin 6, and we'll get granulocyte, monocyte, pollen stimulating factor over here we can get also. We can get the eosinophilic stimulating factor. Okay, we can eosinophil chemotactic factor. Now see, among these are known as what? Cytokines. Whatever the soluble proteins we are getting over here, these are known as what? Cytokines. Cytokines. Now among the cytokines, especially the interleukin 3 and interleukin 5, they can cause further stimulations of the eosinophil. Stimulation of eosinophil. You understood? At first, from the cytoplasmic signals, we will get the preformed products, and these products are not preformed. These will be produced by the what? These will be produced by the nucleus first. Okay, they will be as in terms of the soluble proteins. So these are not the preformed. These will be known as secondary products. Okay. Now we'll talk about another type of the signals that will go from here to the membrane. Okay, let's talk about this thing. Now see, another signal that will go from where? From this from the whenever the Ig is cross binding, after that they will send another signal that will go within the membrane, So whenever another signal that is going within the membrane, whatever the changes we'll see over here, let's talk about them. So whatever the changes we'll get over here, let's talk about them first. Okay. First, now see, whenever the signal is going where, intramembranous signal, whenever you are getting the intramembranous signal, whatever the thing will see within the membrane, this mast cell, they will activate the phospholipase A2 enzyme. And whenever we will see the activations of the phospholipase A2 enzyme, what they will do, they will break down the phospholipids. So we will see the activations of the phospholipase A2, and they will do what? They will break down the phospholipid. They will convert the phospholipid into what? Arachidonic acid. I am writing arachidonic acid. Arachidonic acid. First, see, whenever the intramembranous signal is coming, okay, first we will see there will be activation of the phospholipase A2 enzyme. This phospholipase A2, they will just do the breakdown of the phospholipid and they will convert it into the what? Arachidonic acid. Now this arachidonic acid that can be reacted by the different enzymes. One of the two major enzymes over here, one is cyclooxygenase, another is lipoxygenase. Okay, another is cyclooxygenase. Now whenever the arachidonic acid is going through the lipoxygenase, whatever thing it will produce leukotriene B4, leukotriene C4, leukotriene D4, leukotriene E4. This type of products will be produced by what? Lipoxygenase enzyme. Okay, and whenever the arachidonic acid that will go through the cyclooxygenase enzyme pathway, whatever the thing will get the prostaglandin. We'll get prostaglandin. We'll get the different kinds of the prostaglandin E2, prostaglandin D2, for example, these things. Okay. Now, whatever the functions we'll get over here. Now, okay. Let's use this one. Now, whatever the leukotrienes we are getting over here, try to remember this leukotriene B4. Leukotriene B4, they are the chemical mediators. They are the chemical stimulator for neutrophil. So, the leukotriene B4, they are the chemical mediators, the chemical stimulator for neutrophil. And rest of these things, that means the leukotriene C4, leukotriene D4 and leukotriene B4, they are the most potent vasogenic and spasmogenic factors over here. So, leukotriene B4, they are having the chemical mediators. They can stimulate the neutrophil, and whatever the other leukotrienes in terms of the leukotriene C4, leukotriene D4, leukotriene E4, they are the most potent vasogenic and spasmogenic factor. So they will cause further, they will cause further dilations and the further spastic factor, that means constrictions of the smooth muscles. As a result, the further this type of the problem we see where within these figures. That means the lumen ultimately that will be very narrow. And along with that we will see these things. But these leukotrienes, they can also activate the secretions of what? The glandular cells over here from the glands. As a result, we will see here the mucus that will be produced too much. As a result, we will see the lumen that will be narrowed with the thick mucus. So it will be not possible for the patients to take breath normally. Okay. So as a result, especially we will see where we will see the main problem. The main problem we will see in terms of during expiration. During expiration, the main problem we will see over here. So whatever the treatment we can do in this case is, whatever the treatment, when we are talking in terms of the treatment, we can first, whenever during the emergency conditions, because this is the emergency conditions over here, first we have to give what? Oxygen supply. We have to give the oxygen supply. Then you can use some bronchodilator, for example salbutamol. And the late phase of this thing reactions, the late phase of the reactions, we can use steroids. And if we see, this thing is causing the severe problem that is known as anaphylaxis. So if you see the anaphylactic shock, if we get it over here, the anaphylactic shock, anaphylaxis. we have to use what adrenaline injection. Adrenaline injection, intravenous. Use intravenous adrenaline injection in these conditions. Okay. So this was all about in terms of the the different kinds of the complications and different types of the problems. So we will see these cases and whatever the chemical mediators you are getting in this case, whatever treatments. Okay. So these are all about the type 1. This is a type part 2 video. I have already described the part 1 video about the normal figures. Okay. So thanks for watching. In the next part of the video, we'll talk about the type 2 hypersensitivity in details. Okay. So see you in the next video. Thanks for watching and please do subscribe.